Okay, thank you all for joining me for this virtual presentation. My name is Neil Miller. I'm the Director of Bioinformatics at the Genomic Medicine Center of Children's Mercy, Kansas City. And I'm very happy to be here today to talk to you about some work we've been doing recently on using third-generation sequencing for the diagnosis of rare disease. So just as background, let's remember that there's around almost 7,000 diseases that have been identified that are expected to be genetic in origin. These are called individually rare, collectively common, meaning the incidence of any one disease is fairly low, but taken together, they represent a pretty significant health impact, affecting 1 in 30 children in the US, 1 in 6 hospital admissions, and 1 in 5 infant deaths. And diagnosis in these cases is very challenging, leading to diagnostic odysseys. But of course, there's some significant impact in making diagnoses that can affect treatment and certainly always affects families. The advent of clinical exome sequencing has certainly made a dramatic improvement in our ability to come up with molecular diagnoses for these patients, increasing yields up to around 25 to 35%. In some cases, short-read genome sequencing is able to add to that a little bit. In general, however, we don't see a dramatic increase in diagnostic rates in using genome sequencing, and I suspect that the ones that we do see are primarily due to technical reasons such as reducing coverage artifacts and so on, more than in representing any kind of ability to interpret the gene, the areas of the genome that are outside of the protein coding regions that are in the exome. So recognizing that nearly half or 2/3 of the kids that are going into clinical sequencing are coming out without a diagnosis, the Children's Mercy Research Institute and our Genomic Medicine Center has started a ambitious new program we're calling Genomic Answers for Kids. The goal is to sequence over 30,000 affected patients and their family members over the next 7 years. We plan to create a pediatric data repository of over 100,000 genomes that we want to then make available to the larger research community in order to facilitate the search for answers and novel treatments for pediatric genetic conditions. As enrollment criteria to this program, patients have had to have already gone through clinical sequencing and still come out without a diagnosis. And once they're in the program, we're deploying comprehensive sequencing and novel analytic strategies in order to make the diagnoses that we can. Another word on our open access pediatric data repository. So it's a real key goal of the project in order to make these data available to researchers broadly. And we currently are thinking of this in terms of 3 different access tiers. So the first tier is available currently today. It's completely public, summary level variant data, variant calls and allele frequencies that are accessible through our variant warehouse database. We've also implemented the GA4GH beacon service so that this cohort is searchable from the beacon network. We then have 2 different tiers of controlled access that we're imagining. So we're hard at work right now in establishing a tier 2, which would be available to individual PIs after they've gone through a request process. And this will include de-identified samples containing patient phenotypes, clinical features, pedigrees, and a prioritized variant set in some cases, and then full variant sets available in other repositories. So we're working with the phenotypes group on establishing a prioritized variant data store. We're also working with dbGaP and NHGRI's Anvil to provide access to the complete data using their controlled access mechanisms. And then finally, we're imagining very complete access that would be kind of at an institutional level with some kind of MTA. And we would like to really make data available to partner institutions in some kind of collaborative computing platform, ideally on the cloud. We're employing a really comprehensive sequencing strategy for the project. So all patients enrolled will have exome sequencing, but more commonly will have short read whole genome sequencing. We're generating a variety of functional genomics data, including whole genome bisulfite sequencing and single cell RNA and single cell ATAC. For non-diagnostic cases after exome and genome sequencing, we're selecting individual cases to go into third-generation sequencing. So we initially did a fair amount of 10x genomics linked read sequencing. That product's since been discontinued. But more recently, we've been really exploring the use of third-generation sequencing and long-read sequencing with the PacBio Sequel II instrument. So despite having developed a comprehensive sequencing and analysis approach with short reads, there's still a number of analysis challenges that remain. So the first is with short reads, continuing difficulties with phasing variant calls. And so this has a distinct challenge with looking at Singleton analyses where we just have an affected patient and without parental samples. So this lack of a trio really makes it difficult for us, number one, to identify de novo variants, but also really limits our ability to affect autosomal recessive disease inheritance patterns. Coverage remains an issue. So even with PCR-free genomes, we're still seeing very uneven and even gaps of coverage still that make detecting certain regions of the genome very difficult. We struggle quite a bit still with reading through repetitive regions and regions of low complexity. So this limits our ability to look at some known disease genes that have pseudogenes. It also limits our ability to look at a set of repeat expansion disorders simply because of just difficulties in mapping through the regions and calling those expansions with short reads. Structural variant detection remains a challenge also. So our ability to find large copy number variants and large deletions and duplications is complicated with short reads, as well as the identification of more complex events like inversions and translocations. So in order to explore the utility of PacBio sequencing for clinical rare disease diagnosis, Children's Mercy has entered into a collaboration with PacBio and Microsoft. And as part of this collaboration, we're generating 80 genomes at 30x HiFi coverage using the Sequel II system. And as I talk to you today, as I'm recording this, there's 24 of them done and working their way through our analysis pipelines. We've selected trio cases for these that are negative after exome and genome sequencing analysis. The Microsoft Genomics team is working directly with PacBio to build out Azure analytical capability, as well as a data repository for the cohort that we'll hope to share with the larger community. And so this will take the form of deploying a SmartLink service in the Azure Marketplace sometime in early 21. That SmartLink service can be used in conjunction with the Sequel instrument or as a standalone analysis solution. And of course, we'll use some cloud capabilities to control costs. The lab team is hard at work coming up with some improvements on sample preparation. These are primarily aimed at increasing quality as well as throughput. So there's a novel workflow being developed by the folks at PacBio that's allowing for parallel processing of up to 20 samples at the same time. So comprehensive sequencing requires some comprehensive analytics. So we're using a variety of open source as well as privately developed tools to assess the impact of genetic variants. And then we combine those with patient and family info by using patient symptoms encoded as human phenotype ontology terms and familial relationships defined in standard formats like PED files. We're using automated interpretation tools like Exomizer and Amelie to generate prioritized gene and disease candidate lists that are then combined with the annotated variants to go into a collection of tools that are being used for downstream visualization and interpretation. So this includes tools like Viking and the Variant Warehouse, which are developed at Children's Mercy, as well as open source tools like IGV and UCSC, and then some commercial solutions and a clinical from BioDiscovery, and then of course, trusty Excel from Microsoft. So the analysis of HiFi reads has its own strategy that's different from short reads, of course. So first, circular consensus reads are aligned to the human reference genome build 38 with PBMN2. Small variants are being detected with deep variant in PacBio mode, and then those small variant calls are being phased using the What's Hap tool. Structural variants are being called with the PBSV tool. Variants are being analyzed in parallel using two different workflows at PacBio. There's a tool set developed to annotate structural variants for population frequency and impact on known genes. And then at Children's Mercy, we're using a combination of the ANOT-SV tool and some adaptations to our runes characterization pipeline aimed at evaluating impact of structural variants. Finally, we're doing full de novo assembly using the 30x coverage CCS reads using HiFi ASM. So you can see some statistics of what we're getting from de novo assembly. There's some really interesting possibilities presented by the capability of doing de novo assembly for each sample. So you can see, looking at the assembly statistics, assembly size, and the number of contigs, that we're getting fairly high quality assemblies more or less routinely from these data. And these are being done with computational requirements that really promise to enable this to be done, feasibly, routinely at scale. So these assemblies required roughly 13 hours on a 48-core machine that had, under 128 gigs of RAM. So certainly the server class that's available to a fairly broad number of bioinformatics groups out there. There's some really interesting possibilities that are raised by being able to do this de novo assembly. Initially, what we're hoping to do is explore the use of personalized references to improve sequence alignment and variant calls. So this would be combining novel content from the assembly with the regular human reference genome and then redoing sequence alignment in order to minimize mismapping error. And then a big frontier is going to be to evaluate novel content that's in those assemblies. And I expect there's going to be quite a bit of work we're going to be doing on that in the future. There's some more interesting possibilities if you start thinking about things that are being detected by one technology that are not being detected by another. So looking at a set of samples, 13 samples that we have 30x HiFi coverage, as well as having deep short read and linked read coverage. You can see that in the table on the left, I'm showing that we're detecting roughly 400,000 variants in each sample that are not being detected with another method. On the right, I'm showing that we are seeing relatively good concordance between the PacBio CCS derived variant calls and the short read calls. This is being done in comparison to microarray calls to using that as a gold standard to calculate sensitivity and specificity. And you can see that at a minimum, we're getting equivalent performance, if not in some cases superior performance, from the CCS reads when looked at that microarray, when in comparison to that microarray data. But that does suggest that many of those calls that are being called exclusively in one technology stand a good chance of being real, and there might be some interesting stuff in there. Similarly, structural variants, where our ability to detect these is greatly increased with the long reads, and we're detecting on the order of 10 times as many events with PacBio CCS reads. So when we're doing rare disease diagnosis, one of the key things that we do is compare to reference data sets in order to identify variants that are very rare in the population. And as we start getting into these technology-specific variant evaluations, where we start running up against the limitations of those reference data sets, because most of the very large population databases have not been done with this technology. So sequencing analysis are ongoing, but we do have some initial results to share, especially focusing on 3 of our problem areas. So in the first area of Singleton analysis, I'd like to show you case CMH 1610-01. This is a young girl who presented with a variety of liver problems. She did go into clinical testing, and that revealed a single pathogenic variant for Niemann-Pick disease. Only a single variant though, and this is traditionally seen as an autosomal recessive disorder. In this case, parental samples were not available. Subsequent biochemical testing did lead to the clinical diagnosis. However, a second diagnostic variant was never confirmed in the clinical case. So the patient was enrolled in our GA4K program, and a second intronic pathogenic variant was identified. However, there was still the open question of whether it met the autosomal recessive pattern because the phase between the two variants was unknown. We assumed it was in trans, but it wasn't known. However, so after doing PacBio sequencing, we are able actually to confirm that the variants are not in phase with each other. You can see very clearly, if you look at allele 1, the first variant is here, and the second variant here, and you can see they're very clearly on different alleles. It's important to clarify that while this diagnosis was made with biochemical testing, in many cases, we don't have that kind of fallback method for detecting many of these diseases. And so we're really relying on sequencing to be able to give us as complete an answer as possible. This difficulty is also amplified in very young patients or infants who may not show all the symptoms of a disease. So in our second area of challenge, coverage, I'll show you a patient that was presenting with developmental delay and seizures, and an MRI revealed lissencephaly or smooth brain. Clinical exome sequencing was negative in January of 2020. HiFi sequencing was completed in September of 2020 and identified a pathogenic variant, in CEP85L. In the meantime, between the time clinical sequencing was done and the HiFi sequencing was done, CEP85L, an association to lissencephaly, was published in April. And as we look closer at the pathogenic call, we realized that we were able to make it because the HiFi reads produced quite a bit more even coverage across the region. So if we look at the IGV plot, on the top is the HiFi genome, and on the bottom is the 10x Genomics linked read genome, and there's our diagnostic variant. You can see that the coverage of the CCS reads is much more even, and there's a good representation of the variant across all the reads. It reads in both read orientations, whereas the 10x Genomics call is very low quality. It's in a small number of reads. There's a coverage drop, and the reads are only in a single orientation, making for a very low quality variant call. So this kind of highlights two challenges that we face in doing these kinds of analyses. So the first is in how do we deal with regions like exon 1 that are still remaining very difficult to sequence with exome sequencing. And it looks like the CCS reads are helping us quite a bit there. And then, of course, this also underscores the challenge in how do we stay up with, how do we keep pace with analyses that are changing all the time, and that there's a rapidly changing landscape of disease gene associations coming through. Okay, so lastly, I'd like to talk about some success with dealing with repetitive sequences and repeat expansion. So the CMH1541 family had multiple individuals. We ended up enrolling 9 family members total. They all had some combination of symptoms with different degrees of severity. Clinical testing came back negative. We suspected a repeat disorder. So this was suspected for 2 different reasons. So one, the pedigree really suggested a autosomal dominant inheritance pattern. But also it's known that there's definitely some neurological disorders that are known to be expansion diseases. In the initial analysis of this family, no known disease genes were initially identified. However, a paper came out not very long ago that suggested an association of the STAR-D7 gene to epilepsy, and that led us to take a candidate gene approach for this family. And when we look at the PacBio data for the over the STAR-D7 gene, we were able to find a 5-base repeat insertion of up to 1,000 bases that showed up in the 2 affected individuals. This was then confirmed with triplet primed repeat PCR in other affected individuals. It was also completely absent in the unaffected individuals. And so this tied with the new disease gene association makes for a pretty solid case for a diagnosis. However, the symptoms that are being presented are somewhat novel, and it suggests that we might be actually looking at a novel disease. However, it's more comparison is needed, and then there's a caveat that the reference data sets that we're able to compare to are still fairly limited. So in closing, we're hard at work with novel sample preparation and new analysis pipelines that we've been developing in order to support this HiFi sequencing of human genomes. Our initial analysis of 8 samples has resulted in 1 confirming clinical diagnosis, 1 new clinical diagnosis, and 2 novel gene identifications. Sequencing and analysis are ongoing. We hope to have more to share with you in the future. But we can say that there's some really promising results that we're getting out of incorporating long-range sequencing into pediatric genetic disease diagnosis. With that, I will thank our collaborators at PacBio and Microsoft, as well as all the hardworking folks at the Children's Mercy Research Institute. And I'd be happy to take any questions. Thank you very much for joining me.
Good afternoon, everybody, and thank you for joining us. My name is Nick Cisneros. I'm the Senior Director of the North American FAS team. And with me here is Neil Miller, who you guys just heard speak. He's the Director of Bioinformatics at Children's Mercy Hospital in Kansas City. And this is your live Q&A session. So if you can, please go ahead and enter your questions into your chat box, and I will read those off to Neil here. And we already have a few questions here, so I'll just jump right into it. One of the samples, 2060, had twice the phase block N50 and also higher phasing completeness. Is this due to higher heterozygosity, perhaps mixed ethnicity?
That's a great question, and I have to admit I don't know the sample characteristics right offhand to be able to comment on that, but that's a reasonable assumption, is that we might be looking at a mixed ethnicity. Okay.
And we can look into that a little deeper if there's follow-up on that as well. So one of the next questions is your preference to always do trio sequencing if parent samples are available? And are those factored into the goals of the Genomic Answers for Kids is working towards?
Yeah, absolutely. So we very much are finding lots of value in doing trio sequencing, and the Genomic Answers for Kids program is explicitly trying to enroll family members as much as possible, and it's really to answer a lot of those, to pin down the questions of inheritance, an inheritance mode that I discussed in the talk. But I think hopefully what I highlighted was in cases where we can't get parents, we are actually with the PacBio sequencing able to get a lot of phasing information that does help us answer some of those same questions.
Okay, fantastic. Excuse me. And did you say that you're always doing full de novo assemblies for the examples you showed, or directly calling variants?
We are doing both. So we are routinely doing de novo assembly for all of these samples, as well as a larger set that we're doing that's much lower coverage. I think but we're separately doing your more traditional alignment-based variant calling. And I think we're still working with how to best incorporate the de novo assembly data. So I didn't really have time to get into it too deeply, but I think there's kind of 2 main avenues that we're looking at. One is to use the novel content that we get out of the novo assembly in order to clean up alignment-based workflows and basically avoid forcing mappings to areas in the genome where the read didn't really come from. But then the second is really to start looking at some of that novel content and see if we can characterize it. So if it's not in the reference, it's an open question of what is that content? And then a further question of is that related to the patient's disease at all? And that's definitely going to take some work.
Sure. Okay, fantastic. Next question. Do you think long read sequencing will work well for diagnosing mitochondrial disorders?
That's a good question. So the mitochondria is about 15 kb. I mean, that would fall within the same range as what our HiFi reads are. I think you should expect to get a lot of the same benefits in terms of measuring heteroplasmy accurately with mitochondrial sequencing. I do have to say I'm not a lab guy, and so it's possible that there's some chemistry reason why The mitochondrion isn't quite as sequenceable, but as far as I know, it seems like it should be a good fit still.
Yeah, and there should be no reason that the mitochondria should not, from the lab perspective.
Yeah.
Okay. Next question. Very interesting stories. Were you able to tell the number of repeats ATTTC via HiFi sequencing, and how do you confirm?
So yes, we were able to get a exact number of the repeats, and I think there's some characterization work that we'd like to do with the software in order to kind of put that into the standard molecular diagnostics framework of pathogenicity based on the number of repeats. The one sample I showed, the star D7, I think that was confirmed clinically with, is it maybe triplet primed PCR? I think that's how that was confirmed. But yes, we've been really pleased with the ability to be able to look at the exact motif that's being repeated and get an exact count.
Okay, perfect. And we have time for one more question here. Just for curiosity, how is the sequencing cost covered? Is it covered by the patient, insurance company, or research funding?
In this case, it's all research funding. We're not able, so the only patient funding, we do offer clinical tests, and those are paid for either by a combination of the patient or by insurance. But the PacBio sequencing that I discussed is all purely part of a research protocol. And so that's funded by research funding, both through donors to Children's Mercy, as well as for this project, some donations from PacBio itself.
Okay, perfect. All right, Neil, thank you for your time today. It was a great talk, very good subject, and we look forward to hearing more on this topic as the project progresses.
Great. Thank you. Thank you for having me. I appreciate it.
Yeah. And thank you, everybody, for your time today and listening to our conversation and joining the Q&A. If your question hasn't been answered, we'll follow up with you offline and make sure that those questions are answered. Have a good rest of the afternoon.